Welcome back to our look at muscle physiology and part 4 of our video series on muscle physiology. In this installment, we will be looking at muscle contractions and what are the regulatory mechanisms that really regulate and control that release of calcium that's going to lead to the eventual contraction. And so in some ways we are getting back to the beginning part of our story. We've done the middle, we moved to the end, but we're still missing that beginning section. And so that is where we are going in this video here today. And so the question is, when does that muscle contract? And the answer to that is very simply, the muscle contracts when a threshold has been met. And so when do we meet that threshold? And what is that threshold? Well, the threshold is a minimal voltage across the membrane that leads to an action potential. Now we know, if you remember, we have the sarcolemma. Here's our junctional fold. We're going to come down into that T-tubule there, come back up. We remember that we had coming down into here, you had an axon, and that axon had terminal bulbs. The axon terminal, there's the axon terminal. That there is the sarcolemma. All right, this here is the T-tubule. And so we know that in here we have a release of acetylcholine, which is the neurotransmitter, and that neurotransmitter is binding to sodium receptors or sodium channels. And guess what? Those sodium channels are all along the sarcolemma and the T-tubule. All right. And so what ends up happening here is, let's make a little sodium channel there. Acetylcholine binds to the channel, and it is going to force sodium to come in. And when it does that, you alter the voltage of the membrane. Remember way back a couple videos ago, I said that there's a balance of, you have positive ions on the outside, positive ions on the inside, but one side is more positively charged than the other. This is where this is coming in at. Right? This is where that difference in electrical charge is having an impact. Because as that sodium rushes in, it changes the voltage, and when that voltage reaches negative 55 millivolts, in other words, when there is an imbalance in the electrical charge across that membrane, an action potential is going to be created. There's my AP. And that AP is going to travel to the next sodium channel, which is going to have sodium rush in. It's going to create a new action potential, which is going to have sodium rush in. That action potential travels to the next sodium channel, which is going to cause sodium to rush in, create another action potential, causing sodium to rush in, leading to another action potential, causing sodium to rush in, causing sodium to rush in. And so that sodium keeps rushing in as an action potential reaches that sodium channel. When it happens, a new action potential is created. But again, what is causing that is crossing this threshold of negative 55 millivolts. Right? Crossing this threshold of negative 55 millivolts. One of the things that you are missing here is another term, and that other term is resting membrane potential. Right? And what resting membrane potential is, it's a charged membrane. And this equals negative 70 millivolts. And so a charged membrane is negative 70 millivolts. So as that sodium is moving across the sodium channel, resting membrane potential goes from negative 70 to negative 60 to negative 55 millivolts. And that is when that action potential is going to be sent. So we've got to consider this negative 70 millivolts. That's normal, that's base, that's resting membrane potential. The ions are distributed and the membrane is charged. Acetylcholine comes in, binds, and it allows sodium to start moving, which changes the charge. And once that charge changes to negative 55, boom, the action potential goes. And then it causes this sodium channel to go from negative 70 to negative 55 millivolts. Boom, another action potential is sent. We're going to look on this on the next screen. But it's this that is driving that action potential. If you don't reach threshold, an action potential is not sent, which means the voltage-gated calcium channels are not going to open, which means calcium doesn't get released, which means calcium doesn't bind to troponin, tropomyosin doesn't uncover the myosin binding sites on actin, and a muscle contraction does not happen. So this is critical. This is critical. One complete cycle of contraction and relaxation, one complete cycle of going from resting membrane potential back to resting membrane potential is what we call a twitch. Right? That is a twitch. Now, on this slide, what we are seeing is this cycle kind of laid out for us. So this right here is what we refer to as the latent period. All right. There's tension, but nothing is really happening. In other words, this latent period is characterized by resting membrane potential. Right? In other words, negative 70 millivolts. Right? Negative 70 millivolts. So right here, we have negative 70 millivolts. We're at resting membrane potential. There is tension in the muscle, but the muscle is not contracted. Right? A stimulus comes in. That stimulus is an action potential. And it begins to have sodium crossing the sodium channel. And it will do that until it hits threshold at negative 55 millivolts. And what you're seeing here is a sharp rise in voltage till about plus 50 millivolts. During this rise in millivolts, we have contraction that is occurring. Once you've hit contraction, you then come down this slope into something that is called relaxation. In other words, tension reduces. And what you do is you actually drop down to here to about 80 millivolts, and then you come back to negative 70 millivolts right there. This cycle of latency period, contraction, and relaxation that's happening in the muscle. This is what's happening here in the muscle. All right. So contraction is occurring. This is within the sarcolemma. Right. This is in the sarcolemma. But this is what's happening in the muscle. This is what's happening in the muscle. So as all of this is happening, the muscle is contracting and relaxing. You're going to see a couple other terms that we're going to tie into here. So this is what's happening during the muscle. This is what's happening in the sarcolemma. This is what's happening in the sarcolemma. This is what's happening in the sarcolemma. This is happening in the muscle. This is happening in the muscle. Right? So when the muscle, or I'm sorry, when the sarcolemma goes from this latent period to positive 50 millivolts, calcium is being released. And when calcium is being released, contraction is happening. But when that sarcolemma drops back down to negative 80 millivolts, there is no contraction. And so calcium is being reabsorbed. Maybe that's another good way to look at that. Can I add another color to this? I certainly can. Let's do black. Calcium release. Calcium reabsorption. All right. So what determines whether or not something meets threshold or not? Well, that comes down to something that we call the all or none hypothesis. Right? I like to use the analogy of you're either going to flush a toilet or you're not going to flush a toilet. All right. So whether or not the muscle is going to contract is going to be based off of whether or not there is an action potential. Again, all or none. If you hit negative 55 millivolts within the sarcolemma, action potential is sent. That action potential is going to drive the release of calcium, which is going to allow for the muscle to contract. If you do not hit negative 55 millivolts, calcium is not released. And when calcium is not released, muscle doesn't contract. Right? It's either all or none. You're either going to contract or you're not going to contract. You're either pregnant or you're not pregnant. You can't be kind of pregnant. You either are or you aren't. Muscle's either going to contract, muscle's not going to contract. The toilet is either going to flush or it's not going to flush. Right? If you're in there and you're doing your business and you go and you press down on that handle and you just lightly tap that handle, toilet's not going to flush. You have to push that handle past a threshold. Got to push that down more than 50%. Then the toilet is going to flush. And this next thing is very, very important. If you take your fist and you slam down on the toilet handle, does the toilet flush any more aggressively? No. A flush is a flush. Same thing with an action potential. An action potential, it's an action potential. It's always going to cause the same response, release of calcium. Right? Flushing a toilet is always going to cause the same response. Water is going to leave the bowl, and it's going to get flushed, and whatever's in it, it's going to get carried out with it. Doesn't matter if you just barely flush that handle and push it past threshold, or whether you slam it as hard as you can. Right? So action potentials always are generated at the same strength, no matter how far past threshold you hit. But a twitch is very different. That contraction is very different. You can have weaker contractions or stronger contractions. And what determines that are those 5 things that you see there in front of you. Right? How quickly the action potential arrives. How quickly the action potential arrives. Can go ahead and increase the strength of a contraction, because if you have more action potentials arriving, then you have more calcium being released. And if you have more calcium being released, you've got more calcium available to bind to troponin, which means you can uncover more tropomyosin, which means you can uncover more myosin binding sites on actin, which means you can have more actin binding to myosin to have more contractional strength. This is what happens when you pick up a cinder block. Right? If you pick up a pen, you're not sending that many action potentials because you don't need that much muscle contraction. But if you all of a sudden pick up a cinder block, you have to send more action potentials to release more calcium, to bind to more troponin, to uncover more actin, to allow more actin to bind to myosin, so you can have more sarcomeres contracting. So quicker stimulus, more AP arrival, is going to lead to more calcium present, which is going to allow you to contract more. You ever walk up to something and you're anticipating it to be heavy? And you go and you pick it up with the anticipation that it's going to be heavy, and all of a sudden, whoo, it like goes up in the air. It's like you almost throw it up in the air because it's lighter than what you thought. Why is that? Because your body was overstimulating the sarcolemma with action potentials, which means it secreted all of this calcium, and all of these sarcomeres were now activated to pick up something that you anticipated to be heavy. And it wasn't. So you overpowered it. All right. I'm going to skip the third one here real quick. And I'm going to jump to looking at temperature. Right? Temperature will definitely have an impact on contraction. All right. And how often. So think about in the morning, if you get up and you try to run and you haven't, those muscles are cold, right? It takes a little while to warm up. All right? Cold sarcomeres tend to be closer together, and so you have to work to warm them up. In other words, you have to work to pull the actin apart so that there's not as much overlap, which goes into this. How stretched are those sarcomeres? And we'll talk more about this here on the next slide. Temperature has an impact on how stretched the sarcomeres are. Right? Warmer sarcomeres are more stretched. Cooler sarcomeres have less stretch to them. And so therefore there's not much room for them to contract. Same thing here with hydration and pH. Right? Your water content and the pH of that of your muscles have an impact on that. It almost paralyzes them if the pH is too low. In other words, acidic. So think of lactic acid building up within the muscle, and you get a Charlie horse. All right? That's exactly what we're talking about here. And so keep in mind that when we talk about how stretched the sarcomeres are, right? Here's a thick myofilament. Here's a thick myofilament. You have some thin myofilaments coming in here. 1, 2, 3, 4. Ignore him. Right. This setup, this is overstretched. Right? Because if these guys are binding to there and to there, there's not much for the thick myofilament to grab onto. So what kind of a contraction you're going to have? You're going to have a very weak one. As opposed to this. So if I put another one in here, here's your thick myofilament. Okay, now watch this. The opposite of that is this, where now you've got, here's your thin myofilaments. Those thin myofilaments are really overlapping thick myofilament, and so yeah, you have all of this binding that's happening, but when those myosin heads flex, how much are you really going in? Because remember, you've got your M zone in here, and that's going to block it. So you can't, these guys are too tight. This is like your muscles when it's really cold. This is like your muscles when they're overly warm. Right? And so what's the ideal? Well, the ideal is come up here, you have your muscle fiber. You have a little bit of overlap, you have a little bit of overlap, a little bit of overlap, a little bit of overlap. Now you can have enough myosin-actin binding going on. Right, you've got room, because here's your M line, you've got room to contract and shorten, and so you get an effective contraction. And so that's what we mean by how varies, or varies on how stretched the sarcomeres are. There's something referred to as recruitment. All right, and so recruitment is dealing with how many sarcomeres do we need to activate. How many sarcomeres need to be activated? Well, the more sarcomeres we need to recruit, the more action potentials we need sent, so that way we can release more calcium, so more calcium is available to bind to troponin, so we can spin more tropomyosin to uncover more of the myosin binding sites on actin to allow more actin to bind to more myosin, so you get a stronger contraction. Right? That's recruitment. Right? That's recruitment. Remember that not all sarcomeres are going to be activated at the same time. The reason for that is because all sarcomeres are going to contract and relax. They're all going to go through that cycle that we looked at a few minutes ago. And so you're going to have some sarcomeres that are contracted, some sarcomeres that are relaxing, and you're going to have a lot of overlap of contraction and relaxation within the sarcomeres. But the more fatigue that the muscle experiences, the more sarcomeres they're going to activate to help maintain the contractional force. Again, maintaining. So while some are relaxing, others are picking up the slack to contract. And you maintain the contractional force. We refer to this as TREP. Right? the increase in the tension within the muscle, the increase within the contractional force within the muscle due to repeated stimulation by action potentials. The more action potentials you stimulate, the more calcium is released. The more calcium that is released, the more sarcomeres you can stimulate to increase the contractional force. But there is a limit to this. There is a limit to this. And that limit is so we have what we define as being incomplete tetanus and complete tetanus. Incomplete tetanus means that there is sarcomere contraction and relaxation. Complete tetanus means that there is sarcomere contraction continually. Right? In other words, no relaxation. Right? No relaxation. How does this come about? How does this happen? Well, this happens because under normal circumstances, incomplete tetanus is defined as 20 to 40 action potentials per second. Right? So by sending 20 to 40 action potentials per second, you never reach a point to where those sarcomeres are only contracted. They're always going to be contracted and then relax, contracted and then relaxation. Right? It's estimated that even at full workout strength, we typically only reach about 27 action potentials per second. And that's at full workout. That is pushing your body 100% during a workout. So we don't even come close to having tetanus, right? Or this complete seizing of the muscle. Now, for complete tetanus to occur, you are dealing with anything that is greater than 40 action potentials per second. That will lead to a continual contraction within the sarcomeres. When do we see that? We see that during, if you've ever had electric stim or a TENS unit hooked up, that's constantly firing off at the muscle. That's firing at a rate of more than 40 action potentials per second. It's stimulating the muscle. And so that muscle contracts, it stays contracted, it's tight, and then it backs off and it's allowed to relax. And then that stimulation occurs again, and it goes ahead and it continues to contract. We see complete tetanus with some types of bacterial infection. All right? Botulism, all right? Botulinum, all right? It's a Clostridium botulinum, all right? It's a bacteria. Clostridium tetani, it's a bacterium. It causes muscle seizures. All right? You take the neurotoxins out of botulism, out of the botula bacteria, take the neurotoxins out, and we inject that into people's faces called Botox. And that's what causes them to look like they're constantly smiling, or to wrinkles disappear, or that they can't close their eyes, because those muscles are in a state of complete tetanus. It is not the norm by any stretch of the imagination. Now, just kind of real quickly, where this energy is all coming from, right? Where this energy is all coming from are two specific proteins. All right. Myokinase is one of those proteins. All right. So myokinesis is the process. Myokinase is the protein. Oh, what the heck happened? White, that doesn't work. Myokinase is the enzyme. Right, the other enzyme that we need to be concerned with is creatine kinase. So how do these two work separately? Well, to answer that, myokinase is a protein that's going to regulate the transfer of a phosphate group from one ADP to the next. So in other words, you have ADP, myokinase is going to take a phosphate group from here and add it onto here. And when you do that, this becomes AMP, adenosine monophosphate, and this becomes ATP. All right. This would be an example of substrate level phosphorylation, right, where this protein, myokinase, is taking a phosphate group from one ADP and transferring it to another ADP to create ATP. Right. Creatine kinase does something a little bit different. Creatine kinase is an enzyme that works with a molecule called creatine phosphate. And creatine phosphate has stored phosphate on it. And what the creatine kinase does is the creatine kinase takes from creatine phosphate, the phosphate group, it takes that and it puts it onto ADP to create ATP. All right, so the creatine kinase steals the phosphate from creatine phosphate, adds it to the ADP to create ATP. This tends to be a little bit more effective because here every ADP molecule can be turned into an ATP molecule. Using myokinase, you've got a 50% increase because it takes 2 ADPs to create the 1 ATP. So creatine kinase is a little bit more effective at creating that cellular energy, that ATP. Because remember, this is a energy intensive process. All of that ATP needing to bind to myosin. This is also why you will see creatine phosphate added to post-workout drinks. This is why you will see creatine kinase added to very often pre-workout drinks to go ahead and help to sustain energy creation throughout the workout, creatine kinase in that pre-workout, and to aid in the recovery of the workout. Those post-workouts have creatine phosphate in there, so increase the availability to make more energy to heal as we go through. And so one of the things that we see here is creatine phosphate and creatine kinase is usually used for about 15 seconds at the initial onset of working out. So in other words, it takes your respiratory system a little bit to get up to speed. And so your body will utilize creatine kinase and creatine phosphate to jumpstart cellular respiration until your respiratory system gets caught up. And so it buys about 15 seconds worth of get up and go speed for that. All right. After that, anaerobic respiration will kick in. Right, and that will usually supply enough ATP to keep you going for about 30 to 40 seconds. So after a minute, your respiratory system should be up and going, and you should be delivering oxygen enough that you are sustaining cell respiration for long-term ATP production. We are almost done here, guys. I know we went a little bit longer on this video than what I'd like to, but that's okay. You're doing okay. All right, and so again here, this is just a reminder of phosphorylation, right, and what is happening. How exactly are we creating that energy? And again, anaerobic respiration will create ATP, but it's not as productive. Remember this from chapter 3. And aerobic respiration, of course, is the most productive. It produces the most. And so we start with things like creatine kinase and creatine phosphate. That buys us a good 15 seconds. We move into anaerobic respiration to buy us another 30 to 40 seconds of ATP production at the onset of working out. And then long term, we have cell respiration and anaerobic respiration. All right. You will very often hear that there are different types of muscle fibers. Fast twitch versus slow twitch, and depending on the type of working out you do, you're either activating your fast twitch or your slow twitch fibers. Well, what in the world does that all mean? Well, let's consider. All right. Your slow oxidative fibers, or what we define as being your slow twitch fibers. Right, your slow twitch fibers. What these guys are doing is these guys are using ATP more slowly, right, when bound to myosin. It's still using cell respiration. Right. But because the ATP is more slowly utilized, you are resistant to fatigue. Right? In other words, you can work longer and harder with this. What are we talking about? What activities are using slow twitch muscle fibers? Running uses slow twitch. If you're doing things like wall sits, you're activating more of your slow twitch fibers. Planking, right. Posture, in other words, just sitting or standing. These are all things that are utilizing those slow oxidative fibers, these muscle fibers that are contracting at a slower rate, and so they're utilizing the ATP a lot slower. Fast oxidative glycolytic fibers. Fast oxidative glycolytic fibers, on the other hand, are utilizing ATP through cellular respiration, and they're using them at a even slower rate, because fast oxidative is doing both anaerobic and aerobic respiration. So it is replenishing the ATP at an even faster rate. And so this has a high resistance to fatigue. What are we talking about? We're talking about walking. Right? We're talking about walking. We're talking about sprinting, short bursts of energy. Uses the energy very quickly. It can be replenished through aerobic and anaerobic respiration to maintain it. High resistance to fatigue. in that way. All right. And so that is your fast oxidative glycosidic fibers. And then lastly, you have your fast glycosidic fibers. Not your fast oxidative, but your fast glycosidic. And these guys are have the lowest ability to resist fatigue. And so these guys tend to fatigue very quickly. Quick fatigue. And the reason for that is because they lead to a quick breakdown of glucose during glycolysis. And so it's a quick burst of energy that is being produced. You can do very intense workouts over short periods of time. Intense but short workouts. All right. And this is typically weight training. Right? And so this is where you get big and bulky, right? When you lift a whole lot of heavy weights. You have muscle fatigue. Those muscles give out after a certain amount of reps. You can't sustain bench pressing as long as you can during sprinting or walking. And so muscle fatigue sets in. All right. And just lastly, a couple little bits of information here on smooth muscle. All right. Some differences with smooth muscle here. There are no T-tubules. There's even fewer sarcoplasmic reticulum that are present. And what stimulates that release of calcium is much different since there are no T-tubules. And so what's stimulating the contraction, what's stimulating the release of the calcium are hormones, things like gastrin, things like gastrin, leptin, ghrelin. Right? These are your hormones that tend to regulate muscle contraction. Carbon dioxide, changes in pH, stretch receptors within the stomach can also stimulate muscle contraction. And so there's a lot more variability in how we contract smooth muscle. By the way, calcium, since there's very few sarcoplasmic reticulum, calcium is just available within the extracellular fluid, within the matrix that is surrounding these cells. And the other thing is there's no troponin that is present. And so instead, what takes the place of troponin for binding of calcium is a protein called calmodulin. And calmodulin is found on the thick myofilaments. And so calcium binds to the calmodulin, which then activates another enzyme that we call myosin light chain kinase. And that adds, myosin light chain kinase adds a phosphate group back onto the ATP that is attached to the myosin head. And that's what allows the myosin to then bind to the actin that's on the thin myofilaments. So calcium binds to calmodulin, which activates the myosin light chain kinase, which adds a phosphate group back onto the ADP to create ATP on the myosin head. And then that allows myosin to bind to actin. Because of this very quick conversion of ADP to ATP without the use of an action potential, smooth muscle can sustain contraction for a very long period of time. In other words, myosin can just go ahead and keep contracting multiple times over and over again. And this here just shows you structurally the difference between skeletal muscle and smooth muscle. All right, so you can actually see in the smooth muscle on this side oops, you can see over here that you can see over here, this here is smooth muscle. So in other words, the actin and the thin and the thick myofilaments don't line up like they do within skeletal muscle. This is why smooth muscle is non-striated. It's non-striated because those thin and thick myofilaments are not lining up like they do within skeletal muscle, which is why you see striations. You see that banding where you have the overlapping of the thin and the thick myofilaments. And then this summary table here is just that. It's a summary table of skeletal, smooth, and cardiac muscle. And this is a good review table for you going into that 4th lecture exam, which will be on muscle physiology, and our next PowerPoint, which will look at how we create an action potential a little bit more in depth. And so this brings us to the end of our look at the skeletal system, the muscular system, and more specifically, muscle physiology. I encourage you to go back and review these videos frequently and often. And as we get together for our little chats and Q&A sessions and class time, I encourage you to ask questions. And so with that, I apologize for the length of this video. It's a little bit longer than normal, but I will catch you on the flip side.